So a key defining feature of biological species is that they are reproductively incompatible. So different species don't produce offspring together. For example, these two species of sea urchin, the red and green sea urchin, they live in the same habitats, but their sperm and eggs are not compatible and they don't produce offspring together. And so the evolution of this reproductive incompatibility is central to the process of speciation, but the mechanisms by which reproductive incompatibilities arise are not very well understood. And so for my postdoctoral research, I'm studying the genetic basis for the evolution of reproductive incompatibilities in the red sea urchin. So sea urchins are a very good study system for understanding the genetic basis for how the evolution of reproductive incompatibilities because their fertilization occurs externally. So males are releasing sperm, females are releasing eggs into the water column, and these gametes are floating around until they bump into each other and bind in the process of fertilization. And so this means that we can study how the eggs recognize sperm from their own species and avoid binding with sperm from other species that might be also floating around in that water. So in sea urchins, gamete recognition proteins mediate this compatibility between eggs and sperm. So you can think of these like a lock and a key. Only the correct key will open a specific lock. And so different species will have different forms of these locks and keys that don't match up. Within a species, you can have small changes to the genetic code leading to changes in these gamete recognition proteins that reduce the compatibility within that species. And then in the process of speciation, these changes to the genetic code will accumulate and select for big differences in proteins and really change the compatibility between sperm and eggs. And so my postdoctoral research is looking at the genomic basis of these gamete recognition proteins and understanding how small changes can occur within a species. So to do this, we start with the DNA, with the genetic code that's coding for these proteins, and we identify the genomic location of the genes that code for the proteins. And this DNA is going to be found in all cell types, so it's going to be found in both sperm and eggs, the gene that codes for one of these proteins. But it's only going to be expressed and turned into RNA and then into protein in the cell types where it's needed. And so the DNA is turned into RNA in the process of transcription and then converted into the protein in the process of translation. And so if we look at, for example, the sperm protein, we'd expect that it's only going to be expressed or turned into a protein in sperm and not in eggs, even though the DNA is going to be in both sperm and eggs, the DNA coding for that specific sperm protein. And so in the first 4 months of my postdoc, I've been able to identify the genomic location of the proteins that code for both sperm and egg proteins. So the sperm protein has been pretty well characterized and is a relatively small protein. And so we found the genomic location for this, and we were able to confirm that this gene that we found in the genome codes for a sperm protein because we only found the RNA for that gene in sperm and not in eggs. The egg protein has been a little more challenging because it's a very large protein, and research in other sea urchin species have identified potentially 2 different egg receptor proteins for sperm. And so we've found the gene that codes for these 2 different proteins and identified the genomic location. And when we look at that RNA for one of these proteins, we're detecting the RNA for that gene only in egg cells and not in sperm cells. However, the RNA for this other potential gamete recognition protein is found in both sperm and eggs, suggesting that it may not be the egg receptor for sperm. And so now that we've identified these 2 gamete recognition proteins and confirmed that they're expressed in the cells that we're expecting, we can now look at small changes across individuals within the red sea urchin to understand how that affects compatibility within the red sea urchin to better understand how reproductive incompatibility can arise over time. Thank you.